tonight we have our speaker, Dr. James West, got his, I just asked him how his career evolved. He got his bachelor's degree in range science, Utah, I believe, Utah State. In 1996, in 2002, he got a PhD in botany, and that's a retraining already. He spent some time in Minnesota and back in Utah before he came to Texas A&M. He's now an associate professor in Department of what? Ecology?
Ecosystem Science and Management.
Ecosystem Science and Management, with involvements in forensics and plant science and other environmental departments here. He will talk to us about different isotopes and how they're distributed. His work involves one other thing that's dear to my heart. I was a nerd as a kid, that's why I have the job I do as a chemistry professor. And I not only liked science, but they always knew that I would be interested in an atlas or an almanac, and I loved geography. So our speaker combines a little chemistry and some geography, so I should have a great time, even if you are having any difficulty keeping up. I will hope not. So, without further ado, let me welcome Professor James West to talk to you about isoscapes.
Thanks, Ed. All right, so I guess the first question is, can you all hear me? Especially the folks way up there. No, it's a sincere pleasure to be invited to participate in the lecture series. It's true enough that I've had I guess a diverse career, but it's always been around a fascination with the natural world, and I think chemistry helps us understand that. It helps us probe and study and understand this beautiful, complicated planet that we all live on, and also one that we significantly influence ourselves. So I'm going to talk about isoscapes and kind of explain what that is. For better or for worse, it's a word I made up. And I'll explain kind of what the word means to me and kind of some of the science that it's motivated. And so I have kind of two pictures up here, one that's on the left and that I'm sure you're familiar with from NASA, and then the one on the right, which depicts some kind of gradient. And I've got some symbol down there that depicts something about 2H. So this is an isotope. This is what I've called an isoscape of hydrogen isotopes and precipitation on the planet. And I'll talk about how these things are generated and kind of some of the applications and some of the scientific and other consequences of that variability. But I want to start with a quote here. So this is from 1926 from Vladimir Vernadsky, and he's credited with kind of defining biogeochemistry. So this is a long quote, but I think it's interesting and kind of gives you a sense of how I think about the planet in terms of its chemistry. So life is potently and continuously disturbing the chemical inertia on the surface of our planet. It creates the colors and forms of nature, the associations of animals and plants, and the creative labor of civilized humanity, and also becomes a part of the diverse chemical processes of the Earth's crust. There is no substantial chemical equilibrium on the crust in which the influence of life is not evident and in which chemistry does not display life's work. So this describes what is broadly now known as the discipline of biogeochemistry, and it paints a picture of a planet in which the chemistry, our ability to describe and understand the chemical reactions on this planet can't be done without thinking about the living things on that planet. The chemical reactions that happen on this planet are very different from the chemical reactions in many cases from what we might see on Mars, for example. And the reason for that is because of the living things that exist on this planet. So this is one of those chemical reactions that you might be familiar with. So this is the sort of summary chemical reaction of oxygenic photosynthesis in plants. So we have CO2 and water, and then combined with the energy of the sun, that produces carbohydrates and oxygen, right? So you're all familiar with this. This is the reaction that powers nearly all of the life on the planet. Another thing that can happen, and I'm not depicting a chemical reaction here, but this is, the arrows are showing water, or are supposed to be showing. You're supposed to imagine water flowing through the plant. That's me up in the tree, by the way. So this is depicting water being taken up by the plant from the soils, transported up in through the plant as liquid, and then it finally gets up to the leaves. And once it gets up to the leaves, it changes phase to vapor and leaves into the atmosphere. All right, so this is something we call transpiration. So plants are constantly moving water from deep within soils up into the atmosphere. So this is what we think of as transpiration. Plants, of course, participate in all sorts of different reactions, photosynthesis and transpiration being dominant ones that you're all familiar with. And of course, as a biogeochemist or as an ecologist or even as a climate scientist, I want to understand these interactions. So the interaction between the vegetation, let's say on the surface of the earth, or the crust as Vernadsky might describe it, and the gases in the atmosphere. So we have CO2, water, and oxygen in the atmosphere. CO2 is taken up and released by the vegetation. Water is taken up from soils and released into the atmosphere. It also rains out of the atmosphere. Oxygen is produced in oxygenic photosynthesis. It's also consumed then in respiration by all of the organisms that you can see there. So we can actually study that. We can study the concentration of water vapor in the atmosphere, CO2 in the atmosphere, et cetera, and learn something about those interactions. But in fact, if you're an isotope person, you know that it's not quite that simple. In fact, the gases that are exchanging look more like this. So it's not just one molecule of CO2, it's a variety of molecules of CO2 with a variety of isotopes associated with that molecule. So you have the most common molecule of CO2, C12, 16O, 16O, and then you have a bunch of others with C13 or 18O attached to them. They're all behaving chemically more or less as CO2, but they react at different rates. And those different reaction rates encode information about how CO2, in this case, is getting from the atmosphere into plants, or how, in fact, it might be returning to the atmosphere. Or why is the atmospheric CO2 going up in the atmosphere? And you might be able to trace those processes. Similar kinds of things can be said for water vapor and oxygen. Okay, so I know you all know about isotopes, but I'm just going to level the playing field here. So isotopes are these nuclides with different masses, right? So this is hydrogen with the sort of most common hydrogen with a single proton, right? 99.9816% to 99.9974% of hydrogen on the planet is this. A little bit is this, 2H. So we have atom with a proton and a neutron. And so some hydrogen exists like this. It's not a lot. You can see the values there. But there's some of it out there. And it more or less reacts as hydrogen, but it has double the mass. So having twice the mass means that it diffuses more slowly. It's less likely to go from liquid to vapor phase, et cetera. And this becomes rather interesting. If we can actually measure the distributions of these isotopes in natural systems, we might be able to learn something about, let's say, rates of evaporation or photosynthesis. Okay, so just to introduce the notation, when we talk about stable isotopes, so we have this small range of variability here, and you just bear with me here a minute. We have this small range of variability here, and we have very sensitive instruments that allow us to measure very, very precisely, not the concentration, but in fact the ratio of the heavy to light isotope. So we don't measure the concentrations directly, we measure them indirectly by measuring the isotope ratios. So this is what we're talking about. So we have an isotope ratio. It's just the ratio of how much of the heavy to light isotope you have. And remember that the heavy isotope is the less abundant isotope. That's basically universally true in the isotopes that we're talking about. This is our hydrogen example. So the R value is 2H over 1H. So how much 2H versus 1H do you have in that sample? And then, to make things confusing, no, it's not really to make things confusing. It is a little bit confusing, but it's not to make things confusing. We express it as a delta in a delta notation. The reason we do this is because my instrument, the instruments in my lab, will measure that isotope ratio very, very precisely. But the absolute value I get out of that instrument might be slightly different from the instrument in my friend's lab in Hawaii. Okay? If I want to compare what I'm doing here in Texas to what my friend's doing in Hawaii, we need to be on the same scale. So we measure the ratios very precisely, and then we have some material that's common material, and we all measure that as a standard. You're all familiar with standardization. So in the case of water, what we do is we say, as a community, a planet-wide community of scientists, we say, okay, so here's our big bucket of water, and it's nearly that simple. So we have a common material. I don't necessarily know what its isotopic abundance is, but I know that it's all the same. So what I'm going to do is I'm going to distribute that material to all the labs around the planet, and we're going to define that as the standard. And then we're all going to be on the same scale. So we know that if we analyze our samples this way, that we're all expressing values on the same scale, and therefore those values can be comparable. We do this in using isotope ratio mass spectrometry, and this is just an example. So the instruments that we use measure the isotope ratio abundances of gases. So they're directly, these are gas source, fixed magnet isotope ratio mass spectrometers. But I might want to know what the isotope ratio is of water, or soils, or a cherry. And I have to get the carbon atoms or the oxygen atoms in that material into gas form. So what we'll do is we have peripherals, and those basically convert the organic or liquid compounds that we're interested in into a gas, say CO2 or hydrogen or carbon monoxide, and then that gas is introduced into the mass spectrometer. So that's what you're seeing here. This happens to be a compound-specific instrument where we can look at individual compounds, individual amino acids, individual fatty acids, et cetera. We do that in the lab. So if you go to sibs.tamu.edu, you would go to our stable isotope lab. We're over in the Kleberg Center. You can come visit us if you like. You want to come check out what happens in the lab. Maybe not all at once. Maybe smaller groups. But you're welcome to come visit us. And I and others will make these kinds of measurements in the lab. Okay, before we get to the good stuff, I need to introduce one more essentially vocabulary word, and that's fractionation. So fractionation is simply a word that describes the difference in the isotope abundance of one pool relative to another, and that difference being caused by some chemical or physical process. So you can see up here I've got depicted water molecules. So some of those water molecules have 18O associated with them. Some of them have 2H or 1H. And you can see kind of a mix of what we would call isotopologues of water. So different molecules of water with different isotopic composition. If you take that, assume that's a pool of liquid water, and you then convert it, you go through a phase change to vapor. That phase change is going to favor the lighter isotopes. They have higher vapor pressure. So if you only take some fraction of that, say 25% or something, and you move it over to the right, you create a little bit of vapor. The vapor isotopic composition is going to be different from the liquid isotopic composition. And that's because of the slight differences in the properties of these different isotopologues with different isotopes associated with them. So that change in the isotope abundance from, in this case, liquid to vapor is called fractionation. And we use that word generically. If it's an enzymatic reaction, a phase change, any process that results in a difference of the isotopic composition of the product relative to the source, we call fractionation. One quick point here too. So in your careers down the road, when you start thinking about applying isotopes and doing various things, if I took all of that liquid water and put it on the right and made vapor out of it, okay, I can actually see you, which is good. How many of you think that the vapor isotopic composition will be different from the liquid vapor isotopic composition we started with? If we take all of it, move it from the left to the right. Okay, a few hands going up. So this is a little tricky thing about isotopic composition. Fractionation is only important when you are taking a fraction of that pool and converting it to vapor. So if you take all of the liquid and convert it to vapor, all of the atoms on that side came over to the right side. And so this isotopic composition is going to be identical. So if you looked at that change over time, it will be very different. If you take the first little bit, it's going to be lighter, have lighter isotopes than later on. But if you take it all, it's going to be identical. Okay, so we finally try to get to something interesting. So this is the hydrologic cycle. So that's happening planet-wide at various rates with various isotopic composition starting points. So we have water in the atmosphere. It's sourced mostly from the ocean, but also from the land surface. We make from liquid to vapor. That's a fractionating process. So the clouds are different from the ocean. Those clouds move over the land surface, it rains. That rain is different from the cloud. So this creates an interesting pattern that will I can depict here. So the ocean, so this is our delta notation, 18O here, is 0. And it's 0 by definition, because we use ocean water as the standard. So the ocean starts out at about 0. It's not exactly that. There is spatial variability in ocean isotopic composition, but works pretty well for this cartoon example. You evaporate off of that ocean. The vapor now is minus 13. Now don't get too confused about the minus number. That just means that it's lower than the reference material. It's lower than 0. So it has less of the heavy isotopes in it. That minus 13, and then we express it in parts per thousand per mil. Rains, you produce minus 3. But you've only taken out a little bit, so you've left some behind, but that vapor is now minus 15 because you've dumped a little bit of the heavy stuff out. Move over, and now your rain is minus 5, and then the vapor the residual vapor is also depleted. So this sets up a nice spatial pattern in the isotope ratios of precipitation across the landscape that already could be really interesting to use. We also have plants. So plants are taking up that precipitation because it's falling in the soils and transpiring it back into the atmosphere, but along the way, the oxygen and hydrogen atoms of that water are being incorporated into the organic compounds that the plant is producing. So to a first approximation, the isotopic composition of those leaves is going to be a function of where it is on the landscape, simply by taking that liquid water and making organic compounds out of it. And it will fix this rather fluid, if you will, isotopic composition of the rain into organic compounds that we can also analyze in the mass spectrometer. So this distribution then says, okay, so this is interesting. How repeatable is this? What does this look like at large spatial scales? We've been measuring precipitation for a very long time. So this is some examples of the locations where we've been measuring isotopic composition and precipitation. This is from the International Atomic Energy Agency. Measurements have been going on for several decades, and you can kind of see the distribution here. The data here is from the 1960s. So we've been measuring these isotopic composition around the planet. So what do we see? Well, it turns out that we see a nice sort of, I'm going to actually get this thing here. So we have this, if you plot the oxygen and hydrogen isotopic composition of all of that rain against each other, you get this very nice line here. And this is called the global meteoric water line. And the slope is a function of just the fractionation, and if you're looking at hydrogen versus oxygen. Okay, so the difference in mass between 2H and 1H is much larger than the difference in mass between 18 and 16, which are the two stable isotopes of oxygen. So you get a slightly different slope, but they're really tightly correlated because the process that's causing that variability is the same. It turns out that these are warm places and these are cold places. So the cold places, so this is down to minus 400 or minus 50. If you recall, the ocean is starting out at 0. So cold places are really different from the ocean. And that has to do with air mass trajectories and then also the details of how that fractionation works. But cold things are very different from the ocean, less so than warm things. And you can see this. So this is plotting the same data for oxygen isotope against general temperature. So cold things tend to have low isotopic composition. Okay, so finally, so what? It's the first year program lecture series. It's not just supposed to be a lecture on just chemistry. I'm supposed to be telling you why this is interesting. So here's a question, what do I do with this? Well, here's an example of what you can do with this. So here's an orange grove. And the orange trees are doing what I described. The orange trees are taking water from the soil and they're transpiring it. But some of that water ends up in these oranges along the way. And the water in those oranges isn't exactly the water in the soils. What happens is the water goes into those oranges and then it sits in there and evaporates over time. So it's replenished a little bit by the soil water, but it evaporates over time. That evaporation causes the orange water to diverge from the water that fell from the sky, from the, what we call meteoric water. So that evaporation causes a fractionation in that orange water. Okay, so we have this orange juice. Say, okay, well that's kind of, I'm not sure where he's going with this. So I make, if I'm a farmer, I'm gonna make some orange juice. I have to put it in a bottle. There's a lot of water in that orange juice. It costs a lot of money to ship it around the planet, right? It's a lot cheaper if I just evaporate off some of that water, ship a concentrate, and then add water kind of on the other side of the ocean before where I send it. So, but then I can't say it's freshly squeezed, right? So if I'm a little not very scrupulous, I might try to call orange juice fresh squeezed when it's really concentrate, from concentrate, because it's cheaper to ship. It turns out that if you do that, yeah, so here's my cheat here. I might ship concentrate and call it freshly squeezed. Well, because you wouldn't know if I just get a freshly squeezed orange juice that's labeled, it must be freshly squeezed. It turns out that sometimes people, not often, but sometimes people aren't always totally honest with labeling. And so we can actually detect that with stable isotopes. So here's our friendly global meteoric water line. This dotted line is actually the global bottled water line. And all of you know that bottled water mostly is just local tap water, right? It's not entirely true, but a lot of our bottled water looks a lot like precipitation, which looks a lot like tap water, which also, by the way, also looks like river water. So local water often tends to look similar in terms of its isotopic composition. Anyway, so the meteoric water line and bottled water line are similar. And so that's interesting. And what I was saying was that the oranges are using that local water. And so the orange water doesn't look like just local water. It actually looks very different. What happens is, I told you those oranges are evaporating, right? So what that means is that orange water falls off of this meteoric water line. And the reason it does that is to do with the details of evaporation in a dry or a wet atmosphere. But evaporation in a dry atmosphere causes the water to diverge from that meteoric water line, which means I can take a sample of orange juice and tell you right away whether or not somebody has reconstituted it. So in this case, if you added 100% of the water in that orange juice came from precipitation or local water, tap water, bottled water, then it would fall on the meteoric water line. Authentic, freshly squeezed orange juice falls off of that line. And people are applying these kinds of tools for authentication. Okay, so I think that's kind of cool. You might not. I wrote cool up here because I think it's cool. I haven't told you yet what an isoscape is. So isoscapes, so this is a definition. So an isoscape is a spatial distribution of isotope ratios. I've already showed you a map, right? And this global meteoric water line is a function of the spatial variability in isotopes. But generally here, an isoscape is thought to be a model product, and it's derived from isotope landscapes. So this is the idea, is that there are landscapes of isotope ratio variability that we can understand and maybe use. Okay, so we think these can help us answer a few questions. And I'm going to pose a few here and talk to you about some of the work that I've been involved with and others have been involved with as well, applying isoscapes to a variety of questions. And I've picked a few examples that aren't that similar to each other to kind of demonstrate the breadth of application. I know that a lot of you have a wide range of interests, and so I'm going to try to cover a few different topics that you might find at least one of them interesting. So the first one I'm going to talk about is how does trade in illicit materials work? And then I'm going to talk about a slightly different question. Who has a larger effect on savannas, termites or giraffes? And then a third one here was, who was this Jane Doe? Okay, so maybe think about that. So we'll start with illicit materials. So we could pose this question. So this is something you see in the news all the time. There's a raid and the law enforcement discovers a variety of things in this location, and lots of this stuff is not legal. And so it might be interesting for those folks, for the law enforcement, to understand where some of that stuff came from. They might be interested to know that. So isotopes maybe can help us provide information, and on the sort of trade in these things. It's not so if you're trying to figure out how the trade in methamphetamines or marijuana or whatever works, you could go ask people. They might not tell you their sources, right? Probably don't. But you can actually get some information from the material itself. So we'll go back to this. So we have this network. So I told you we made these observations and we discovered these sort of globally coherent patterns that make sense and are temperature dependent. We can take that data and create a model. So this is a model. So this is showing you absolute latitude. So this is just distance from the equator. And these are precipitation 018 values. And you can see, as you go far away from the equator, those values drop. And they drop also as a function of elevation. So again, cold places, more different from the ocean, lower isotope ratios. I can fit a curve to describe that based on latitude and altitude and write that equation. Now, I can apply that equation at every spot on the planet because I can describe the latitude and altitude of every spot on the planet. And I can produce what we would now refer to as a precipitation isoscape. So to the extent that that equation describes reasonably well the precipitation, this map describes reasonably well that isotopic composition of that precipitation. And if you're super motivated, you can go to isomap.org, so as a web page that we created using funding from the National Science Foundation, and make your own isoscape. So you can make your own modeling decisions, your own map. So that's one example. Okay, so again, let's say we believe that this describes reasonably well the precipitation, and that then also translates to the water that plants have access to, so we have this pattern here. And in this case, now we're interested in a particular plant. This plant is not now a tree that we're interested in. This plant is a marijuana plant, let's say. So we said, okay, so we're interested in that, and this was part of a project that I did when I worked on when I was there. All of the samples that we obtained were obtained legally through a laboratory in Mississippi. And so actually when these seizures happen, they go to a lab in Mississippi and they do chemical analyses on them, and then they would ship us a small amount. Of course, law enforcement were involved. We have to weigh the samples when they come in and weigh the samples when they go out, and they were in a locked safe behind an alarmed door. It's slightly different working on this than working on pine trees, I have to admit. The fundamental chemistry is, of course, the same. So this is our map of precipitation isotopes. And in this case, we were interested in gridding that and binning it into a small number of bins because we knew there was noise around our observations of marijuana. And then this is where the marijuana came from, distributed around the country, and we were dependent on the samples that were available. So some portions of the country were better sampled than others. Okay, so we're interested now in then how does this precipitation signal translate to the marijuana itself? And so here's kind of just I'm not going to show you the mathematics of the models, but just to give you a sense of how we do this, we have a water source map that I already showed you. In this case, we generated another one for the United States. And we have a model that we think describes how plants are converting that soil-water isotope ratio into an organic compound. So again, I'm not going to bore you with the details of this model, but we have a physiological model or an observationally based model that describes how the precipitation relates to the plant composition. This model is a function of climate, so we have climate as an input as well. So we run this model driven by the climate and source water isotopic composition, and we produce this what we call steady state map of the isotopes of marijuana. And that's what this map is. So this is a predicted isotopic composition of marijuana isotopes around the planet. We've compared this with authentics and done some double-blind examples and discovered that the model behaved reasonably well. And then we moved on to say, okay, so then can we take this model? We now think we have a distribution of isotope ratios of marijuana around the United States. And then what we'll do is we say, okay, we have this model, and we will apply that to these so-called retail seizures or street seizures. So again, it's much like the introductory slide. We have material, we don't know where it came from. Can you tell us something about it? And so at each one of these locations, we have a number of samples going into this group, and you can see some interesting patterns, I think. All right, so the pie charts are here described as divided into eastern United States, so the origin should be eastern United States, north of the US border, south of the US border, or western United States. And So you can see some interesting, is Los Angeles different from San Diego, perhaps, closer to the border with Mexico. You get even closer with, or conceivably there's some differences between Phoenix and San Diego. You move further north and yeah, perhaps not surprisingly, the material that's being seized in Seattle, we see no evidence that it's coming from south of the border, but there's quite a bit of evidence that there's significant movement across the Canadian border in the north. And you can see some other patterns as well. So this kind of information is being applied by law enforcement agencies in terms of trying to understand these markets, and we've argued that it might be interesting to do this on a sort of time frame. So now we've done this. This is prior to legalization, right? And folks are familiar with certain states have made recreational use of marijuana legal. So you would expect that to change this market. It's still, various aspects of it are still illicit, but you might expect that that action would change the market. And so we could go back and use our marijuana isoscapes to try to better understand how this market is behaving now that the laws have changed. So that's one example. How about a different one? So here's a different question. It says, so our question here is, how have grasslands responded to climate over the past 20,000 years? So an entirely different system, a very different question, in a very different time frame. So going back to 20,000 years ago, we're talking about the last glacial maximum, and that's what this picture is here. So you can see the Laurentian ice sheet here, dipping into the United States, covering a good portion of North America. Over that time period from 20,000 years to the present, so this is 22,000 years ago, so this is denoted as the last glacial maximum. So this gives you a sense of the CO2 concentration in the atmosphere, and this is the proxy global temperature for that time period. So over this time period, CO2 concentrations went up and temperature went up. So perhaps relevant to today, the question is, how did grasslands in North America respond to that change? And we argue that isotopes actually can help us understand that. So I have to tell you one thing about grass biology before we can get to thinking about using isotopes to answer this question. Plants have, well, most plants have two dominant photosynthetic pathways. There's an additional one we won't talk about today. There are C3 plants and C4 plants. Most plants are C3 plants. So the first product of photosynthesis is a 3-carbon compound. For C4 plants, the first product of photosynthesis is a 4-carbon compound. So that's why they're called C3 and C4 plants. C4 plants effectively have a CO2 concentrating mechanism. I won't get into the details too much, but they increase the CO2 concentration associated with a primary enzyme associated with photosynthesis. Those different pathways, though, have really dramatic consequences for the carbon isotope ratios of plant tissues. So we have this nice tool. C3 plants that use this kind of mechanism have this distribution, and it nearly completely doesn't overlap with the distribution for C4 plants. What this means, though, is that we can now measure the isotope ratios of these plants, or the organic compounds in soils, or even of the tissues of animals that eat those plants. And then be able to say, oh, well, it was mostly C4 or mostly C3 or some mixture of both. So some folks, this is not my work, this is work of some colleagues at the University of Utah and other institutions, took advantage of this distribution of C3 versus C4 plants to say, I think I can reconstruct what the vegetation, or at least the grass vegetation in North America was doing over the past 20,000 years by sampling extant and fossil bison. And here's this little quote here. It says, "You are what you eat." There's a fractionation associated with conversion of the organic matter in the grasses to the organic matter in the fur or other tissues of this animal, but it's relatively small and fairly well described. So we can use bison tissue to reconstruct what that bison was eating, and you can also use fossil woolly mammoth. And so what they did was they got access to tissues from living bison and then fossil bison and woolly mammoth to try to reconstruct the grassland distributions in North America. So this is their, oops, pushed the wrong button there. Sorry about that. This is the distribution, and so the open circles are modern. The red ones here are from fossil. You can see that they're distributed around North America. So again, it's bison and woolly mammoth tissues. From those observations, they were able then to reconstruct the vegetation of North America over the last 20,000 years. So what's depicted here is on the top, this is the measured carbon isotope ratios of the tissues that they looked at in these fossil and living animals. And those are then interpolated, so they used a model that allowed them to predict the isotopic composition of the diet, basically, in between the observed observation points. And that then, based on the carbon isotope composition, you can see the purple stuff is close to -12, and the yellow stuff here is close to -27. And if you remember the distribution, C3 plants are close to -27, C4 plants are close to -12. There are more details in the model as well, but suffice it to say that they use the model to then predict what the percent C4 grasses were in the past. So this is modern, mid-Holocene, so 5,000 to 7,000 years before the present, and then the last glacial maximum, somewhere on the order of 20,000 years before the present. So it's a little bit difficult to see in the figure, and so there's this additional figure that shows you the difference. So this is mid-Holocene to modern, so the last 7,000 years or so, and then this is the full period from the last glacial maximum to the modern. And what you see is that the C4 grasses, these plants that have the CO2 concentrating mechanism, actually increased over that time period. CO2 concentrations were increasing over that time period, and based on just their physiology, you would expect the reverse. You would expect that the plants that are investing in the CO2 concentrating mechanism would be disfavored in a high CO2 environment, that higher CO2 concentrations should favor C3 plants over C4 plants. In fact, they see the opposite. They see an increase in C4 plants over time, and they argue then that although CO2 concentration should drive this shift towards C3 plants, there was a C4 shift, and that's likely a result of the change in the climate, and not so much temperature, but a change in the distribution of precipitation. So more precipitation in the summertime tends to favor C4 grasses. And so they argue that because of the shift in the distribution of precipitation, the climate over the last 20,000 years favored an expansion of C4 grasses. So this is interesting in terms of trying to understand the past, but these tools now might be able to inform what we might expect in the future. As the atmospheric composition changes, how will the vegetation respond? And it is a consequence of the atmospheric composition and climate. And so there may be surprises in some ways in how the biosphere responds to an anthropogenically changed atmosphere. Okay, so that's an interesting one there, I think, as well. What about a slightly different question? So here in this question, we're asking, where was this person before they died? So this is another sort of forensic question. And it's very common for, unfortunately, for law enforcement to find bodies or deceased people in locations and have no idea who they are. Sometimes using typical forensic tools, they're able to reconstruct a story, to reconstruct what happened to this person, to identify who they are. Some cases you can use genetics tools, right, to identify who they are. But in order to use the genetics tool, you need to know, you need to have a sample to match the sample to that you might find associated with that person. So you need to find out if there aren't a match, if you don't have a match, you don't have a relative, many cases, these cases will go cold. And so they will basically come to the end of their ability to identify who this person is and say, we can't do any more. We really are not able to identify who this person is. This is where, in some cases, isotopes can come into play. And so again, this is our isoscape here of local tap water. And just like plants convert tap water into organic compounds, animals, like humans, take that water and incorporate it into the organic compounds in their hair, let's say the proteins and the keratin in your hair. And so what happens is the person consumes the local water and the isotopic composition of your hair and tissues associated with that person then can reflect the isotopic composition of the water that that person is drinking. So there's a picture here. It's just a young boy drinking, and this conversion of the drinking water then to an organic compound is embedded in a model here. Again, I'm not going to bore you with the details of the model, but there are models that describe how the biology of these organisms, humans in this case, convert the drinking water into the isotopic composition of the proteins in their hair, for example. And so if you know what that transfer function is, if you know how the organisms convert water into organic compounds, you can produce then a predicted isoscape of what you would expect the hair to look like. So this doesn't tell you, it doesn't solve the original problem that I posed, but it does tell you that there's maybe a tool here in your hair to reconstruct where this person was before they died. We know that hair grows at a particular rate, which means not only do we have a record, a spot record of where this person was, but you might actually also have a record of travel. So that if the person moves from one location to the next, if they move, let's say, from somewhere in Texas out to Nevada and then back to Texas, you should be able to see that record in their hair, similar to tree rings, if you're familiar with tree rings. So this tool was applied to one example. So this is a person who was discovered by duck hunters in the year 2000 near Salt Lake City, Utah. It's called Saltair Sally because it was near the Saltair. It's a resort that is on the Great Salt Lake. Unfortunately, duck hunters found this body, and the law enforcement were unable to figure out who this person was. They had very little information, and they were unable to match this person with anyone in the database they had access to, and really had no idea where she came from. So this case then went to some researchers at the University of Utah, and they looked at the isotopic composition of her hair. And so that's what they're showing here. So this is the O18 of the hair, going from closest to the base of the hair out, and so this is going back in time. And what they saw was this variability in the isotopic composition of the hair, suggesting that she had moved. She wasn't in one location for the entire time period that this covered. This length was converted to an estimated amount of time based on our understanding of the biology of hair growth, and a model that can convert the isotopic composition of the hair to what's in the body. So we have body water, and that's actually the water that's being used to produce the hair, and therefore the isotopic composition of the water that's being used to produce the hair. And so then so we have this initial model. Now there's a little subtle thing here that I'll just point out. Since the person is moving back and forth, once you move to a new place, it takes a while for the body water to turn over, for the compounds in your body that are producing hair to turn over, and so they have to do an additional assessment here to estimate what the signal would be under equilibrium. So recognizing that she's moving around, and she's moving around fast enough that the hair never really comes into equilibrium with the body water, but there's an additional modeling tool that allows them to estimate the equilibrium isotopic composition had she stayed in each of those locations long enough to reach equilibrium. So this is the important punchline here, and what we can do now, if we have this isoscape of hair, we can then take what we think is the equilibrium signal and identify likely regions of where, if you have an equilibrium signal of this isotopic composition, where in the United States, in this case, might you have lived and produced that hair. And so you can see that it's here we've got this region here, and then kind of moving perhaps west, a smaller region, and then down to this perhaps region 4, further south, then back up here, and then over maybe further west, and then again back to here. So this includes parts of the Northwest and Utah and even the Midwest. If you take kind of a simple, so this is identifying a variety of regions, so even into the Midwest. So you might take a simple assumption and say, well, this person's moving back and forth on the order of, a few months here. I might, to a first look, I might assume that the person is moving relatively short distances. So I would take something that's relatively parsimonious with all of these locations, and I might isolate myself to a region like this. So we know we found her in Utah, so that she was found here, consistent with this body water signal. So she was found here, so she had been spending time apparently in that location, which anyway is consistent with the isotopes. And so you might say, oh, there's you know, if we're incorporating all of these maps, I suspect that she might be spending time in this region. And so what that allowed them to do was to point to the northwest, and in this case they pointed to Washington. And they were able to link that to a missing persons case. So what they were doing is they're identifying locations outside of Utah where, you know, do you have a missing person? Is there someone who's identified as missing and you haven't been able to figure out where they went? They were able to link it to a missing person case there, and so now they have a new set of DNA materials that they can compare to this, this Jane Doe, this person they couldn't identify. They've used DNA then to link her to her mother who lives in Seattle, and were able to positively identify her then as this woman, as Nicole Beckles. So they were able to point law enforcement into a direction and then successfully link using DNA this person to her mother who lived in Seattle. So those are three examples that I wanted to talk to you about and share with tonight. I will say that we're here, and like I said, it's an open invitation, again, to come visit the lab if you're interested in stable isotopes. And if you're interested in following up, again, on what's here, I'll point you to two papers. So this is the review, and then the paper on the vegetation change. Thanks for your time. And if you have questions, I'll be around. I'm used to a smaller lecture hall. Yeah, thank you.